In this video, we'll once again be looking at this picture of a puggle. Actually, we're going to be looking at the proximal convoluted tubule, the PCT. So in the previous video, we talked about the glomerulus, this whole section here. Now, we're going to be talking about this dark green section, the proximal convoluted tubule. Proximal because it's the closest segment to the beginning of the nephron. This is the beginning of the next nephron. So this is the closest segment. Therefore, it's called proximal. Convoluted. Convoluted means to be bendy or kind of wavy. Kind of like this. It's wavy. It's convoluted. Okay? So it's the proximal, wavy, or convoluted tubule. Tubule because it's simply a tube. Okay? So this is the segment we'll be talking about. So just a quick recap of what goes on here. So here we have this segment, right, is the glomerulus. And we know that our blood arrives at the glomerulus by the afferent arteriole because A for approaching, remember, afferent for approach, efferent for exit, E for exit. So the afferent arteriole brings our blood. Our blood has good and bad things in it. So as this good and bad things approach the nephron, now it gets put into these small capillaries. These small capillaries are very permeable, meaning that things that are small enough can leave, okay? So, what will leave our blood then? So if our blood arrives here, what's gonna leave? So this is what it looks like. So this here is the membrane that we look at here. So if your blood arrives around here, we're looking at this segment. So right here would be inside your blood. This would be the layer. This would be the layer, the capillary layer, so the membrane of this capillary. This would be the green layer called the basement membrane, and this blue part would be called the podocytes. That's the last layer. So whatever's in your blood, so let's take this molecule, whatever gets here, or any part that gets any part into here, can leave if it's small enough. So let's use this molecule. If it's in here, it can pass the first layer pretty easily, but it can just barely pass the second layer, but it can still pass. So if it's small enough, it can exit your blood into this space called the Bowman's capsule, okay? So basically, in a nutshell, the glomerulus sorts your blood by size. So everything that is small enough can exit, and everything that is too big, such as this guy right here, will stay in the blood, okay? So it's pretty much just a size filter. That's all it does. If the component of your blood can cross these three layers, it'll exit the blood. Okay, so now all the things that can pass. So for example, in this instance, in the last video, we saw that these things can pass. These four things. Okay? Everything else, so these big things, stayed in the blood. They're too big. And we noticed the red things represent bad things in our blood, and the green things represent good things. So this segment does not separate good and bad. So it doesn't keep the good things in our blood and make the bad things pass through. All it does is it separates by size. So both good and bad things can be filtered. Therefore, all these good and bad things will enter the next stage, which is the proximal convoluted tubule, this green segment. Okay? So we know that we don't want these good things to be filtered out of our body, because if it doesn't go into our blood, it means it's going to leave our body, right? If these good things continuously stay in this tubule all throughout the nephron, then it means it will leave our body through our urine. Okay, so we want to see what's going to happen next, because ideally we want to keep these good green things in our body, and these red things we wanted to leave our body. So let's see what happens in the proximal convoluted tubule. Okay, so we're going to be looking at this segment. So by the way, this is very important. So you notice how the efferent arteriole leaves, right? It will have blood in and whatever wasn't filtered, because the blood comes in, things are filtered, some things are not filtered, and it exits. So the things that are not filtered, the blood that is not filtered will exit by the efferent arteriole, okay? This arteriole, I'm going to emphasize here, will continue on. So it's going to continue on like this, and see, it can kind of goes next to the proximal convoluted tube. So this is what we're going to be talking about. You can transfer some of the things from the proximal convoluted tube into the blood if you want to. So if you have some good stuff here that your body wants to keep, it will be transferred back into the arterial, okay? So let's see how this happens. So we're going to be looking at a segment like this. The bottom, so this here is the green segment. So this is the proximal convoluted tubule. And then we have the vessel, the artery above, okay? So this artery will have all the non-filtered stuff. So everything inside it, all of these things were not filtered. You can see all these things, they were not filtered. This one was too big to be filtered. Maybe these were just happened to miss the hole and they didn't get filtered. Okay, so we got the non-filtered stuff. And then at the bottom here, we have the things that was filtered. These things were small enough. And the blood is flowing, I mean, these things are flowing this way. As you can see by the arrow, they're flowing to the right, okay? Now, we want to keep the good things. So what the proximal convoluted tube does is it's going to absorb the good things. So let's look at these. These are little cells, okay? You can see the white nucleuses. These are cells. And you can see they have small little hairs called microvilli. These microvilli, I don't know if you've learned before, but they help to absorb things. They make it easy to absorb things. They increase the surface area to absorb molecules. So let's look at this. So what they will do is they will take the good molecules and they'll put them in your blood. So let's take all these green ones. They'll in your blood. So first, this good molecule will be absorbed into the cell. Then the cell will pump it out into the blood. Okay? Well, first the cell will pump it out into the space, and then it will go into the blood from there. It will diffuse into the blood. So again, here, this molecule, the microvilli takes it up into the cell puts it into the blood. Oh, let me move this to front to make it clear. Okay, so that's moved into the blood vessel now. And last one. Okay, this one I also got to move to the front. Okay, so now we absorbed all these good things, okay? But we made sure to keep the bad things inside the proximal convoluted tubule, okay? Because we don't want to bring these back into the body. Now, some small details that you need to know. You need to know that these good things were brought into the cell and into the bloodstream by active transport, okay? Let me show you here. Active transport means, let me show you, well, let's first of all talk about this. So I'll come back to active transport in a second. So 90% of the filtrate, so filtrate is basically the things that isn't here, the things that were that were filtered from the previous stage in the glomerulus. Those are called filtrate. 90% of the filtrate will get reabsorbed into the bloodstream by the proximal convoluted tube. So almost everything, because a lot of good things will filter and we need to bring back all those good things. So 90% of the filtrate gets reabsorbed. Very important. Now, we know some of the good things that managed to get filtered included glucose, water, ions, and amino acids because they're all very small. So they were able to get filtered really easily through here. Now, these things are all reabsorbed, and they're reabsorbed by something called active transport. Active transport means that these molecules were absorbed against their concentration gradient. This means that, for example, let me take this. Let's say this is glucose. The cell has a lot of glucose. So if you want to take up this glucose into the cell, it will be against the concentration gradient. It will be against the flow. So this is going to require ATP or energy. Okay? So bear this in mind. Everything except one thing that I'll mention just now is absorbed by active transport, meaning you need energy in the form of ATP to take it into the cell against the concentration gradient and then into the blood. Okay? Active transport. This means one important thing. Active transport means you need energy. And we know energy is made by the mitochondria. So you need to know that these cells have a lot of mitochondria compared to normal cells because they have to make energy the whole time to be able to absorb all these important molecules. So for example, the normal amount of mitochondria could be like 100,000 in a normal cell. So that's a crazy amount already. Now, these cells would have more than that, even more than that, so that they can do their job properly. Okay? Now, finally, so what is absorbed? I mentioned all the glucose will be absorbed because we need glucose. And again, against the concentration gradient. We also absorb ions such as sodium, magnesium, potassium, calcium. So right now you don't need to worry too much, but ions are important for various small functions that are actually also big functions in our body, but don't worry about this now. You'll learn about what ions do more or less later as well. So just know ions are also absorbed into the bloodstream by active transport. Now remember, I said only one thing is not absorbed actively, and that is chloride ions. So chloride ions would just simply, so pretend this is a chloride ion. It's not, but let's take a good, chloride ions are good. So let's pretend this is a chloride ion. It will be absorbed passively. This means no energy is required, and it can just diffuse into the cell, no energy, and into the blood, no energy. Okay? So everything except chloride ions are absorbed actively. And lastly, we know a lot of water was able to pass because water molecules are also small, so they are also part of the filtrate. Now, water molecules don't get pumped into the cell and then pumped out. They also flow by osmosis. Now, what allows osmosis to happen? So osmosis happens when, let me show you. So water moves from here into the blood by osmosis. Osmosis is what happens when the cell is very concentrated. So we know this cell has a lot of glucose and things like that make it concentrated. This allows water to flow into it or in between the cells. So it can flow both in between the cells or into the cells to kind of dilute the concentration. And as it does so, it ends up being transported into the blood because the blood is also very concentrated. So water can flow into the concentration gradient to kind of dilute things. Okay, that's osmosis. So that's it. That's all that happens in the PCT, the proximal convoluted tubule. So now you might think, okay, we just removed all the good things and put them back into the blood. So what's there next to do? So next you need to know, sometimes bad things are still in your blood. So they were not filtered. For example, this one missed a hole and this one missed a hole as well. So we want to put these things in here as well. So later on in the next stages, I'll show you how these molecules are transported from the blood into here. Okay, so that's kind of the stage that we're still missing. Okay, but there's still some important other stages that we'll talk about in the next videos. But I hope this made sense.